May it please your Royal Highness, I shall present my lecture on the journey to antibody pharmaceuticals. When we write up our work or discoveries, our accounts are usually simplified. They're written from the perspective of hindsight, and they're trimmed of extraneous detail. They rarely acknowledge the real background, the dead ends, the changes in direction, or the personal, emotional, commercial, or even political factors driving discoveries or applications. In this talk, I'd like to tell you about how antibodies emerged as pharmaceutical drugs, the science and inventions behind them, and how they came to be commercialized, including some of those extraneous details not usually mentioned. I hope you'll see by the end that commercialization is far from straightforward. But let me first set the scene. Until recently, most pharmaceutical drugs were chemicals, reflecting their origins in plant extracts or de novo chemical synthesis. But this dominance was not to last. The invention of recombinant DNA technology in the early 1970s allowed the production of recombinant proteins and development of safe and new medicines based on proteins. The first such drugs emerged in the 1980s, recombinant human insulin for treatment of diabetes, and recombinant EPO and GM-CSF to stimulate red and white cell recovery following chemotherapy. And more recently, the anti-diabetes and weight loss drugs based on engineered versions of GLP-1. By the mid-1990s, we start to see the development of recombinant antibodies for targeted therapies. Of the protein pharmaceuticals, recombinant antibodies are now the largest class. They're now used to treat millions of patients with previously intractable and fatal diseases, and currently generate sales of several $100 billion per year. Now, I was lucky enough to witness and also to participate in this antibody revolution from around the mid-1980s to the mid-1990s. I was based at the MRC Laboratory of Molecular Biology in Cambridge and started my work on antibodies in the early 1980s. I'd just been tenured as a staff scientist, as an independent principal investigator, and reported to Cesar Milstein, the inventor of mouse monoclonal antibodies, a Nobel laureate who was the head of my division and a great mentor. I developed further antibody technologies and became involved in their commercialization, including the development of two therapeutic antibodies, alemtuzumab, now marketed for treatment of remitting relapsing multiple sclerosis, and adalimumab, marketed for treatment of rheumatoid arthritis, Crohn's disease, among other autoimmune inflammatory disorders. Now, in many ways, the emergence of antibodies as pharmaceutical drugs isn't surprising. Antibodies are nature's own pharmaceuticals, created to order by the immune system against the agents of infectious disease. The antibody is a Y-shaped molecule, as shown on the slide. Binding of those antibody arms to antigen may be sufficient to confer a useful therapeutic activity, for example, the neutralization of a virus. But antibodies also have therapeutic features built into their stem, such as the binding site for a recycling receptor, which greatly extends their half-life in serum. The stem also has binding sites for receptors that act as a flag to multiple host defense mechanisms. The abilities of antibodies to block pathogens, to circulate in the blood for weeks, and to kill pathogens by diverse methods accounts for their ability in combating infectious disease. But it's actually their modern application to non-infectious diseases, such as cancer and immune inflammatory disorders, that has made recombinant antibodies the paramount biologic. These modern antibodies bind to human self-antigens, and they work by blocking the binding between protein ligands in human serum and their corresponding cell surface receptors, and by killing the cells that bear the target receptors. The first attempts to develop antibodies as pharmaceuticals started with von Behring, who discovered antibodies in 1890. He set up his own company in Germany, Behring-Werke, using the proceeds of his Nobel Prize to develop horse and cattle antiserum for treatment of tetanus and diphtheria. But the animal serum, which I've marked as number 1 on the slide, provoked an immune response, which on extended or repeated treatment led to life-threatening immune complexes and what was called serum sickness. There was another hurdle. Antiserum comprises many different antibodies and differs from one immunized animal to another. For the development of pharmaceutical products, it's necessary to make a single species of antibody, i.e. a monoclonal antibody, with a defined activity. In 1975, Köhler and Milstein at the MRC Laboratory of Molecular Biology discovered how to make such monoclonal antibodies from immunized mice by creating a hybrid cell, or hybridoma, that secreted a single species of antibody. That's number 2 on the slide. The cells could be grown in large-scale culture, allowing the manufacture of large amounts of pure mouse monoclonal antibodies. But as with horse antiserum, the mouse monoclonal antibodies were seen as foreign by patients, and it proved impossible to make human monoclonal antibodies the same way, especially for those binding to self-antigens as required for the treatment of non-infectious disease. Humans are tolerant to their own proteins. They're tolerant of human self-antigens. And the immune system, in fact, eliminates antibodies that encounter self-antigens at an early stage in the development of the antibody response. Fortunately, the invention of recombinant DNA technology allowed the development of antibodies that were tolerated in patients. The first approach involved the fusion of the antigen binding domains from a mouse monoclonal antibody to the effector domains of a human antibody. That's number 3 on the slide. These simple chimeric antibodies are 2/3 human. I came up with 2 alternative solutions. The first solution was to humanize the mouse antibodies by transplanting just the antigen binding regions from a mouse antibody, marked in red, to a human antibody, white, by recombinant DNA technology. That's number 4 on the slide. These antibodies can be up to 95% human. This work led directly on to my second solution, to make 100% human antibodies. That's number 5 on the slide. I created libraries of billions of different human antibody genes, cloned these into a bacterial, virus, or phage, and then selected those antibodies with binding activities to the target antigen, as I shall explain in more detail later. Now, other approaches to make human antibodies have been developed since, in particular making mouse antibodies from transgenic mice which have transplanted human immunoglobulin gene loci. But that is somebody else's story. There are now over 50 therapeutic antibodies on the market, including chimeric, humanized, and human varieties, and hundreds of monoclonal antibodies in development. They're used to treat a range of diseases from cancer to inflammatory disorders, and even from high cholesterol to migraine. 35 years ago, there were none. It's been a revolution in medicine. Now, this slide highlights a few of the recombinant antibodies that have been approved by the US FDA. It includes alemtuzumab, a humanized antibody that kills T and B cells and is approved for treatment of chronic lymphocytic leukemia and relapsing remitting MS. There's trastuzumab, a humanized antibody that kills breast cancer cells bearing the HER2 antigen and is approved for treatment of such cancers. There's infliximab and adalimumab, chimeric and human antibodies, respectively, that bind to and block the inflammatory actions of the protein TNF and are approved for treatment of rheumatoid arthritis, Crohn's disease, and plaque psoriasis. And there's pembrolizumab, which binds to and blocks the PD-1 receptor and thereby activates T cells and is approved for treatment of several cancers, including melanoma. Now, my tale includes the development and commercialization of two of these therapeutic antibodies, which I've highlighted in yellow. The cell killer, alemtuzumab, the first in class of humanized antibodies, and the blocker, adalimumab, the first in class of human antibodies. But the tale also has a bearing on the development of the entire class of therapeutic antibodies. I'd like to start the tale in 1975, when Cesar Milstein published his key paper, which describes the generation of monoclonal antibodies against sheep red blood cells. To Cesar Milstein, this method opened up a host of possibilities, including the use of monoclonal antibodies in diagnosis and therapy. Now, to understand the story, I need to say something about patents. Patents give an inventor a commercial monopoly over the use of an invention or allow the inventor to license others for use of the invention. The invention might be a process or a product. To be granted a patent, the inventor writes up his invention and applies to the patent office. If the patent is granted, it usually lasts for 20 years from first application. If the inventor made the invention in the course of his employment in a company or an academic institution, the patent will usually be owned by his or her employer. To obtain a patent, an invention must meet specific requirements. The patent must be novel. The invention must be new, meaning it hasn't been publicly disclosed or used anywhere in the world before the patent application is filed. The patent must be non-obvious. In other words, it mustn't be an obvious modification or combination of existing technologies or knowledge. It should involve an inventive step that would not have been apparent to a person with ordinary skills in the relevant technical field. The patent must have utility. It must have a practical purpose or a use. It should be capable of being used or made in some kind of industry, not just a theoretical idea or concept. The patent should have patentable subject matter, so certain things like laws of nature, natural phenomena, and abstract ideas are not patentable. And the patent must have a sufficiency of disclosure. It must be described in enough detail that a person of ordinary skills in the relevant field could make and use it. So having told you about patents, I'll go back to Cesar Milstein in 1975. Before publication, Cesar asked the Medical Research Council, his employer, whether they wished to file a patent on his work, which described the method for making monoclonal antibodies. He wanted to publish his groundbreaking work, and if a patent was to be filed, this would have to be done before publication. If he didn't file or left it until after publication, it would be impossible to get a granted patent, as the work described in the patent would not be novel. In fact, at that time, the MRC had a general policy not to file patents. They believed it wasn't right to file patents on medical inventions, as this could inhibit the take-up of new medicines by industry. But in 1948, the UK government had established an organization, the National Research and Development Corporation, or NRDC, to transfer technology from the public to the private sector, including the filing of patents on any inventions of commercial value emerging from UK universities and publicly funded bodies such as the Medical Research Council. The MRC was therefore required to consult the NRDC on any patent filings. In fact, the MRC was not allowed to make patent applications itself. This had to be done by the NRDC. So Cesar sent off his letter and waited for an answer from either the NRDC or the MRC about whether they wished to file a patent on his invention. But answer came there none. So Cesar, aware of the MRC's general policy not to file patents, and worried that somebody else would beat him into print, went ahead and submitted his paper for publication. It seems that the NRDC had seen no reason to file a patent. Even a year later, the NRDC stated in a letter to the MRC, "It's certainly difficult for us to identify any immediate practical application which could be pursued as a commercial venture, and is not immediately obvious what patentable features are disclosed in the Nature paper." As the value of the technology became apparent to everybody but the NRDC, the failure to patent monoclonal antibodies developed political dimensions. The Prime Minister, Mrs. Thatcher, blamed both the MRC and the NRDC. Astonishingly, some elements of the scientific establishment blamed Cesar for publishing in haste, including an influential report of the Royal Society in 1980 on the commercialization of research in the United Kingdom. In any case, the failure to patent monoclonal antibodies highlighted the more general political concern that the United Kingdom was failing to commercialize its science and was missing out on the potential of the new biotechnology. In response to this, in 1980, the MRC and NRDC came together with private funders to create a new biotechnology company called Celltech. The NRDC was deprived of its rights to all biotechnology discoverers in UK universities and the MRC, and these were assigned exclusively to Celltech for a 5-year period. Now, the newly fledged Celltech focused on recombinant DNA technology and antibodies. It set out to make recombinant antibodies inside yeast cells. And in the US, its competitor, Genentech, set out to make antibodies inside bacteria. In both cases, the antibody protein was insoluble and formed inclusion bodies. Nevertheless, and in many people's view, surprisingly, both companies were granted broad claims to recombinant antibodies, in particular for the production of recombinant antibodies comprising immunoglobulin heavy and light chains produced from a single host cell. In fact, as the Genentech and the Celltech patents had similar claims, this led to a patent interference action between the two companies, and the dispute lasted for years, in the end resolved in the United States, at least in favor of Genentech. In the meantime, other scientists in the UK and the US showed how to make folded and functional recombinant antibodies. They used mammalian host cells. Instead of directing the antibody to be produced inside the cell, directed it to be secreted into the culture medium. This made a huge difference. That process was commercially viable and became the main way of manufacturing antibodies for clinical application. Yet, those antibodies were deemed to fall under the claims of the Genentech and Celltech patents. This shows the advantage of early patent claims, even though, in the view of many, those patents teach nothing useful. In fact, this highlights that life isn't always fair, and that's certainly true in the granting of patents and the legal disputes following grant. In fact, the level of unpredictability of patent litigation and the huge costs mean that even if you hold a good patent, you may find it difficult to enforce against rich companies that infringe it. It was at this point, in the early 1980s, that I entered the field. I'd been interested in the origin of life. I wasn't really interested in antibodies, and I was interested in how enzymes had first evolved as life crawled its way out of the primeval soup. I suspected that enzymes must have evolved from protein pockets that fortuitously had weak catalytic activities. If so, I thought this should be a general property of protein pockets, so perhaps I could create some entirely novel pockets and see whether I could find any catalytic activities. At that point, my eyes alighted on antibodies. The antigen binding domains seem to have an ideal structure for creating protein pockets. These domains are located at the end of the antibody arms and comprise a region of beta sheet surmounted with 6 loops. The beta sheet differs somewhat in sequence between different antibodies, but not so much as the loops differ. It had been suggested that these loops of hypervariable sequence were responsible for binding of antigens, and the beta sheet structure was essentially a structural scaffold for displaying the loops. As I mused on the kind of loops I would need to generate pockets, an entirely different thought came into my head. If antibodies had the scaffold structure proposed, then it might be possible to turn mouse antibodies into their human counterparts by simply transplanting those antigen-binding loops from the mouse antibody to the human antibody. This was such an exciting revelation that I abandoned work on catalysis and set about creating a model humanized antibody to see if this was correct. It took us over a year to build the construct, and to our delight, the humanized antibody bound to the antigen with similar affinity to the parent mouse antibody. But although the paper was published in the top journal Nature in 1986, the reception was lukewarm. After all, the humanized antibodies still contained about 5% mouse sequence. Would they really escape recognition by the patient's immune system? So I thought I'd better make a genuine therapeutic antibody and started working with a clinician. His name was Dr. Herman Waldman in the Cambridge University Department of Pathology. Now, he'd already made a rat monoclonal antibody that killed human white cells, potentially useful to deplete white cells in transplanted organs or for the treatment of lymphoma. But that antibody, as had been expected because it was a rat antibody, had raised a blocking response in patients. Well, we found that our humanized version killed human cells in the test tube just as well as the original rat antibody. But we now needed to find the first patient. It was a lady in her 70s who had non-Hodgkin's lymphoma. Her spleen was grossly enlarged with tumor cells. She was neutropenic and dependent on red cell transfusions. Other treatments had failed, and she was expected to die. After just a few days of treatment with the antibody, there were no adverse reactions. There was some regression of the tumor, and I was told the patient wished to see me. I found her knitting. I asked her how she felt. She said she felt good, unlike after chemotherapy. She then asked me how I felt about her treatment. I said, frankly, I was relieved we hadn't killed her and glad the antibody seemed to be working. She said, You scientists seem to be much more direct than doctors. Perhaps you'll tell me how long I've got left to live." I said, "I got no idea." In fact, none of us had any idea whatsoever. We didn't even know if the humanized antibody would be rejected. Perhaps it would be a few weeks, a few months, or a year or more. To my surprise, she said firmly, 2 months is enough. She explained that her husband was dying. It wouldn't be long, and she wanted to be with him when he died. And her comments completely shook me to the core. Because I'm not a hardened medic. They're used to dealing with these emotional crises all the time. I was just a scientist, gone in, and just, it shook me. In fact, the effect was so profound that I made the decision to change the entire focus of my work towards practical clinical application. In fact, the lady was lucky. The antibodies largely destroyed the tumor over the 30-day treatment, and they were not blocked by any immune response against these humanized antibodies. It was the first use of a recombinant antibody in a patient and was the first to show a therapeutic effect. After 1 year, the lymphoma came back and she died. In fact, she might have lasted longer if we'd had any antibody left to treat her. I realized I'd need to work with industry to manufacture drug product. This couldn't just be done in an academic setting. Well, Herman Waldman and his colleagues continued to seek clinical indications for the humanized antibody, which became alemtuzumab, and to find an industrial sponsor. In fact, in the end, the commercial rights changed hands through a series of companies, from Wellcome Biotech to Leukocyte to Millennium Pharmaceuticals to Genzyme to Sanofi. And it's now used for treatment of B-cell chronic lymphocytic leukemia and relapsing remitting MS. I moved on to new antibodies, new technologies, and new challenges. Before our work with Herman Waldman, I'd applied for a patent on the humanizing technology on behalf of the Medical Research Council. But to my dismay, the head of the MRC administration based in London decided to license the patent exclusively to Celtech on the grounds this was required under the Celtech founding agreement that I've already mentioned that was signed in 1980. But further investigation revealed that agreement had just expired. This was 1985, and it had just a few months earlier expired. When we pointed this out, the head said, well, nevertheless, he was still sticking to his original decision that Celtech would have it. Now, I actually completely disagreed with this. I believe that giving a commercial monopoly to a single company would strangle the rollout of humanized antibodies from other companies. Many companies already had some potentially useful mouse antibodies in their freezers. If we could only just humanize those antibodies, it could create a new wave of drugs. Why should Celtech be allowed to tie up the humanizing technology, a technology in which they'd no hand in developing? This could choke off the application of the technology. And surely, as a publicly funded body, the MRC should be encouraging the application of its research for the wider public benefit. In those days, I was very idealistic. In fact, Cesar Milstein and I both thought the patent should be licensed out in a non-exclusive manner, with easy terms for licensees, similar, in fact, to the licensing of the Cohen-Boyer patent on recombinant DNA technology by Stanford University, which had promoted the uptake of gene technology by industry in the late 1970s. Well, these arguments led to the director of the MRC-LMB, Sir Aaron Klug, a Nobel laureate also, invited by this administrative head of the MRC to discuss the matter with him and the CEO of Celtech. Aaron Klug believed in details, and I had to brief him for a whole day, and he put together a whole bundle of papers and notes, and he went down to London to discuss the matter. When he got there, the head of administration told him this was not a negotiation, no detail was needed, that he just expected them to reach an agreement on the principle of the matter like gentlemen. The principle appeared to be that the head had decided to license Celtec. That was that, and we must cooperate. Now, I just couldn't accept this, nor did I believe that such an agreement would survive independent scrutiny. I therefore asked a friend who was well connected in government how to secure such scrutiny. As the MRC relies on the UK Treasury for its annual allocation of funds and has links into the civil service in several government departments, I wondered if I could raise the matter with someone there. But the friend advised that the civil service would not be independent. They would likely support the head of the MRC, but that the Thatcherite government ministers were more likely to support my view if framed in the language of free markets and competition. The friend advised that what I needed to do was to create a crisis so that it would rise beyond the level of the MRC and the civil servants, and it would rise to the level of their political masters. In fact, he pointed out the MRC and civil service would probably do anything to avoid it coming to the attention of ministers, especially to Mrs. Thatcher, who'd blame them all for the failure of monoclonal antibodies. Well, good luck, said the friend. You better go and create a crisis. So I had to think, how does one create a crisis? I'm just a scientist, you know. I'm not used to creating crises. Does one go to the press? Was there time? Perhaps it would be signed, the agreement might be signed the following day. I didn't know. So I agonized, and then I wrote a letter to Celtec, warning them, as the inventor of the patent, I would destroy my own patent rather than let it fall into their hands. I wasn't exactly sure how to destroy my own patent, but doubtless could find a way. And anyway, I just had to persuade them that I might find a way. After sending the letter, I went to see Cesar Milstein, and I confessed what I'd done. Now, I say I confessed, but it wasn't a full confession. I was somewhat what we call economical with the truth. I never told him this action was a deliberate attempt to provoke a crisis. I think Cesar put it down to my youthful idealism, and he was horrified, but I think he was also quite impressed. Anyway, in the meantime, Celtec contacted the head of the MRC. He exploded and instructed the head of the institute, Aaron Klug, to fire me for insubordination. But after a briefing from Cesar, Aaron refused to do so, saying it wouldn't solve anything, which was true. Now, fortunately, the head of the MRC was due to retire later that year. That was 1987, and the crisis came to the attention of his successor. This man had a background in industrial research, and he actually agreed with our suggestion for a non-exclusive licensing policy. He did not want to inherit a crisis or to have to face Mrs. Thatcher. And he decided to hammer out a compromise agreement. So in fact, my friend in government had been right. The crisis had done its work. So the compromise agreement is basically as follows. Celtec were especially interested in making antibodies for the treatment of septic shock. At the time, there was a lot of biotech interest in this indication, and the top targets were TNF or endotoxin. Accordingly, the compromise agreement envisages that Celtec would have an exclusive license for the field of TNF antibodies. They could get on and they could make their commercial products in that, but that everything else could be licensed out to other people for very easy terms, namely low upfront payments of about £25,000 per license and low royalty rates on products between 1% and 2%. The MRC was also allowed to offer the Celtec patents on two-chain expression to its licensees. I had to agree to help MRC licensees work the technology. And I was told if I did all that, I could keep my job, but I had to accept a written warning about my outrageous behavior. In essence, it was a victory for me, but dressed up as a compromise. Now, that agreement had both pluses and minuses. On the minus side, the agreement blocked the development of humanized anti-TNF antibodies by companies other than Celtec. And the development of Celtec's humanized anti-TNF antibody Cimzia, which is a pegylated Fab fragment, was very slow. It was only approved in 2008 for treatment of Crohn's disease. On the plus side, the wide licensing policy and the co-packaging of the humanizing patent and the Celtec two-chain expression patents turbocharged interest in the humanizing technology. People could see a way through these patents whereby they could get on and make their products. One such licensee was Genentech, and it developed several humanized antibodies, including the blockbuster drug trastuzumab or Herceptin for treatment of HER2-positive breast cancer. Furthermore, the new head of the MRC also set up an MRC Industrial Collaborative Center for contract services in humanizing antibodies. One such contract was with Organon, and that led to pembrolizumab, otherwise known as Keytruda. This was later sold to Merck and is now the world's top-selling drug. Another contract with Chugai led to Actemra, another blockbuster drug. So far, the MRC has received over $2 billion in royalty income over the lifetime of my patent and its contract services. That agreement also led fortuitously to our next project, the development of human antibodies, the founding of my first biotechnology company, Cambridge Antibody Technology, or CAT, to the phage display of antibodies, to the development of the anti-TNF antibody adalimumab, and in fact to my Nobel Prize, as I shall now explain in more detail. Our paper on the development of the humanized antibody alemtuzumab and those wide and easy licensing terms had indeed attracted the attention of biotechnology companies. And I was asked by several of them to consult or collaborate on humanizing their mouse monoclonal antibodies. One of the rate-limiting steps proved to be the preparation and cloning of complementary DNA comprising the antibody heavy and light chain genes of the mouse hybridoma cells. To simplify the process for our licensees, I decided to try PCR amplifying the antibody genes from the hybridoma cDNA. Now, everyone knows what PCR is now. It sounds easy, but I can tell you it was not then. Those are the very early days of PCR. For example, we had to design primers that would amplify most mouse variable region genes without knowing most of the gene sequences. We had to make our own thermostable Taq polymerase, and we had to build our own PCR machines. But then I thought, why are we starting from hybridomas? Why not just amplify the cDNA from the spleen of an immunized mouse, create an expression library of the encoded antibodies, and then screen the library for binders? That way, we could bypass the need to make hybridomas. But then, if we could do that, why not start with the cDNA from just human peripheral blood? That way, we could make human antibodies directly and bypass both mice and humanization. Furthermore, we could generate novel pairings of heavy and light chains, potentially allowing us to create antibodies against self-antigens. Now, for example, if we take two different human B cells and copy the antibody variable region genes by the polymerase chain reaction, we can expect to get two heavy chain genes and two light chain genes. If we now link those genes together at random by PCR, we'll create 4 different heavy light chain gene combinations encoding 4 different antibodies. 2 will correspond to the original antibodies. The other 2 will encode new antibodies with new and unknown binding activities. If we now extend this example to 1,000 different B cells, for example, the number of plasma cells in a milliliter of blood, we could in principle make 1 million different antibodies, of which 999,000 would be new. As both the heavy and light chains were entirely derived from human antibodies, the encoded antibodies would be fully human. Furthermore, having stepped outside the biological constraints of self-tolerance, they could potentially bind to human self-antigens. Clearly, if such libraries of antibody genes when they're expressed, they could provide a huge and diverse library of human antibodies against many possible targets. So, of course, we filed patents to cover this invention. But we expected the binding activity, or any particular binding activity, to be low. We therefore needed a powerful selection technology. In late 1988, we became aware we had a scientific competitor in the United States of America who had also thought of making antibody libraries. It was Dr. Richard Lerner at the Scripps, and he was working with the biotechnology company Strategene. Their combined resources were such that I knew I would soon be outgunned, even though we were ahead at this point. But the MRC refused my request for more space and posts to meet this challenge, possibly because I'd been a very difficult person, I'm not sure. But anyway, they weren't very helpful at that point. I therefore wondered about setting up a collaboration with a company, much as Lerner had done with Strategene. But when I went and explained my new ideas to large companies, they weren't interested. Their attitude was, this is blue skies research. What's wrong with humanizing? If it ain't broke, don't fix it. So I decided I just have to set up my own company for this purpose. I'd no money, but did manage to secure funding for a startup to be known as Cambridge Antibody Technology, or CAT, through a friend, Dr. Gregory. He was the founder and director of Peptech, a small Australian biotechnology company specializing in peptide synthesis in Sydney. He was interested in developing antibodies against TNF and, like Celtech, was interested in their use for treatment of septic shock. Peptech had hoped to humanize their mouse anti-TNF antibodies, but then they discovered this was ruled out by the MRC's exclusive license for the TNF field to Celtech. But in fact, our new library-based approach offered a way around that, because we'd be able to make human antibodies against human TNF. It looked like we held some of the key intellectual property and technology for this process. Now, I think it's also fair to say the Australians saw their stake in Cambridge Antibody Technology as much a gamble as a strategic investment. As well as their research on peptides and TNF, Peptech had developed slow-release GnRH implants for horse breeding, and most other members of Peptech's board were associated with the racing industry. Jeff told me that one of the board members had said of my proposal for the seed investment, "Oh, just give the boffin some money and let's see how he trots." I began to realize that our founders were not investors, they were gamblers, and I was the horse. At the same time as talking to Peptech, I'd been talking to a team of antibody engineers at Amersham International who were due to be made redundant as part of a cost-cutting program. The leader, David Chiswell, offered his help with the startup, and he became the managing director. We then squared the proposals with the Medical Research Council, and an agreement was put together very fast and within a few months. The MRC licensed our patents to CAT, and gave me permission to work with CAT in return for MRC shares in the company and royalties on products. In early 1980, a scientist from Amersham, John McCafferty, joined my laboratory to develop methods for the mass screening of antibodies from our libraries. Our choice was inspired by the work of George Smith, who developed a powerful method for selecting small peptides of filamentous phage according to their binding activities. Instead of peptides, we applied it to antibodies. By appending the antibody genes to the coat protein of a filamentous bacteriophage, that's a virus that just infects bacteria, we could make the phage display an encoded antibody on the surface, as is shown in the slide. We could then capture the phage with the antigen binding activities on a solid phase coated with antigen. So if you pass this down a column of antigen after washing and then elution of the bound phage with high or low pH buffer, you can then grow up the selected phage in bacteria. And this can be subjected to further rounds of selection. So in fact, a selection factor of 1,000-fold in 1 round becomes 1,000,000-fold in 2 rounds and 1,000,000,000-fold in 3 rounds. So with this incredibly powerful selection process, we could isolate extremely rare binders, as rare as 1 phage in 1,000,000,000. In fact, with this technology, the same antibody library, the same tube of phage, just a little test tube, could be used to isolate phage binders against one target after another. The antibody genes could then be harvested from those phage and rebuilt into complete antibodies for expression in mammalian cell culture. As you would expect, we filed patents on that invention. I came to realize we developed a form of Darwinian evolution. In fact, it essentially mimics key aspects of the immune system. By using huge populations, that's billions of phage, and ferocious selection pressures, that's 1,000-fold, applied to a very diverse population of antibodies, our process for making antibodies took days instead of taking thousands of years, as is more typical for the evolution of proteins in nature. Effectively, we'd sped up evolution. We started to use phage display technology to make human anti-TNF antibodies for peptech. But then in 1992, a human monoclonal antibody, not against TNF, but against endotoxin, that was being used for treatment of gram-negative sepsis, failed in clinical trials. And that failure blighted the development of all antibodies for treatment of septic shock, including anti-TNF antibodies. Suddenly, the whole field of septic shock, everyone walked out of it. And Peptech terminated its program with us. But within 2 years, a new possibility had emerged for anti-TNF antibodies. It had been discovered in London that you could use anti-TNF antibodies for treatment of rheumatoid arthritis. So when we were approached by Bob Kamen of Knoll Pharmaceuticals to develop an anti-TNF antibody for treatment of rheumatoid arthritis, we were free to offer our phage library approach. And that led ultimately to Humira, or adalimumab, the first human antibody to be approved for therapy by the FDA in 2002. Now, although adalimumab was originally developed with Knoll Pharmaceuticals, that was purchased by Abbott Laboratories. It's therefore AbbVie that markets the antibody. For many years, adalimumab, or otherwise known as Humira, was the world's top-selling drug. In fact, since first marketing in 2005, the total sales of adalimumab have exceeded $230 billion. That's actually more than any other drug in history. Small molecule drugs, antibodies, anything. Even the COVID drugs, it's more than that. So, as for the Australians, the Australian gamblers, they sold their shares sometime after Cambridge Antibody floated on the London Stock Exchange in 1996, and they made a multiple of their original investment. So the gamblers won their bet. And I shared the Nobel Prize in Chemistry in 2018 for the development of phage display technology. So all ended happily, at least for some, in this scientific and commercial venture. In conclusion, the development of therapeutic monoclonal antibodies was shaped by technological inventions, certainly, but there were many, many other factors, including patents, failures to patent, the politics of Thatcher's Britain, a battle between an autocratic administrator and a renegade scientist, a non-exclusive licensing policy, intense scientific competition, a Cambridge startup company, a group of Australian gamblers masquerading as investors, and the quest for anti-TNF antibodies for the wrong clinical application. Those factors had a huge effect, directly and indirectly, on shaping the development and commercialization of therapeutic antibodies. So if you now read some of those key scientific papers, you may see them in a new light. And if you're unlucky enough to have a disease for which antibodies are the answer, then you can marvel at the tortuous process by which they became commercialized as pharmaceutical drugs. Thank you very much.